We are live with Dr. Ben Albensi in our series that we're calling Meet a Scientist. Hopefully, this time we actually are live, Ben. We've had a few technical difficulties. Thanks for waiting, everybody. But I see over on another screen over there, I can see our smiling faces. This is our weekly live stream series that we're calling Meet a Scientist. You can see we've got our guest scientist with us here today. We're coming to you live from the Youth BIOlab at the Albertson Research Center here in the St. Boniface Hospital campus. This is Treaty 1 territory and the traditional territory of the Anishinaabe, Cree, Ojibwe, Dakota, and Dene peoples, and it's the homeland of the Métis Nation. In this weekly live stream, we're going to have conversations with people from around our research center here and learn a little bit more about the work that they do, and hopefully actually just learn a little more about the people here who actually do the science, who actually do all the things that go on in all the labs above me here. We've got Angelica over here watching the chat and just making sure everything's running smoothly. So if you do have questions, Ben and I are welcoming you to jump in and get involved and learn a little more for yourselves. So we're joined today by Dr. Ben Albensi. He's got a really long title. I'll go through it all. There's lots of confusing words here, but he's a principal investigator in the Synaptic, Plasticity, and Cellular Memory Dysfunction Lab in the Division of Neurodegenerative Disorders. And he's also a professor in the Department of Pharmacology and Therapeutics in the College of Medicine at University of Manitoba. Did I get all that right, Ben?
Yeah, that's good.
You're our first guest from our Division of Neurodegenerative Disorders here at the Research Center. We had our executive director in last week, but we thought we were going to rotate through all of our different research divisions here, our different groups of labs, so people can get to know a little bit more about what goes on. So, like I said, you're part of the Division of Neurodegenerative Disorders. It's taken me a lot of years of practice to be able to say that without tripping over my tongue. Can you tell us, like, what is neurodegenerative? Like, what are we talking about when we're talking about neurodegenerative disorders? What are you a part of?
Yeah, sure. So good afternoon, everyone. Thanks for having me, Steve. It's really my pleasure to be here. So what you're talking about really is brain function and nervous system function. And so we're interested in how nerves work. But in particular, we're interested in how nerves get screwed up, how they die, especially in brain disease like Alzheimer's and diabetes or stroke and these sorts of things. So that's really what the title refers to. It's easier just to say D&D, though.
Yeah, exactly. So really, when we're talking DND, we're talking people studying nerves. And when we say, like just for people out there watching today, when we talk about nerves, like I think people think about, getting on your nerves or things like that. And we kind of know that nerves are involved in, our senses. So we use nerves when we're touching things or tasting things. When you say nerves as a scientist who study these things, like what are we actually talking about? Where do we have nerves in our body and what's the big idea?
Well, that's exactly correct, Steve. So we have nerves everywhere. I mean, we think of the brain as being really the hub or the center of all this nervous activity, but we have a nervous system and nerves going throughout our entire body. And in fact, if we didn't have nerves in our fingers and our legs and our organs, we wouldn't feel anything. We wouldn't feel pain. We wouldn't feel sensation. We wouldn't feel the experience of pleasure. So, the nervous system is just like the electrical wiring in your house or in your office. It really conducts the electricity. It sends the message around to different parts of the body. And not only does it send messages, but it receives messages. So our eyes and our ears and our taste buds and all that, it sends messages to the brain and other parts of the body. So it's really one of our major communication systems.
And so as a scientist who studies nerves, like I know upstairs in the DND, we've got some people who really focus on those sensory neurons, the things that we use to touch and feel. But there's others, like a lot of your work looks at some of those nerves in the brain, right? So there's also, I mean, really that's what your brain is, just packed full of all these nerve cells with different jobs.
That's exactly right. So over the 25 years or so, I've been interested in neuroscience in general and brain function. And really what got me interested in a lot of this was left and right hemisphere brain function, but also memory and intelligence. And that's really how I got started in my interest in the brain. is really looking at how different parts of the brain worked and what it controlled, and how do we make ourselves, why are some animals smarter than other animals, or why are some people seemingly smarter than other people? And so those sorts of questions that really intrigued me many years ago as a kid and as a teenager.
So you say you've been at this for 25 years. You're a professor. You run one of our labs upstairs. So as a professor, like you probably have a whole bunch of different jobs. Like we went through your title there. You run a lab, but you also do some teaching. Like what is kind of your job here at the research center? How do you do what you do?
Yeah, so that's one of the things I really enjoy. Being a professor, really being able to wear many hats. So in fact, in the summer I wear this hat right here, and in the winter I wear this hat. When it's really, really cold in January, I wear this hat, right?
Perfect.
So,
That's a good Winnipeg, Manitoba hat. Yeah.
so just kidding, but really it allows me to play a lot of different roles. And so what I mean is that I teach. I'm on teams here, not only locally in Winnipeg and Manitoba, but in the United States. I'm on a variety of teams. I'm the editor-in-chief of a major journal called Molecular Neurobiology. So you really get to do a lot of things. I mean, you get to travel to different meetings in Europe and the US. You get to host meetings here. Of course, we do outreach, and you're great at doing outreach. It's such an important part of what we do at St. Boniface is to run the Youth BIOlab. So, and we do that here. We give tours that you conduct and to other groups. So you really, really get to do a lot of different things as a scientist and as a professor working here or working at a university.
And I think that's what a lot of people out there watching think when you think of what a scientist does. We really just think of someone working at the lab bench, using like the tools and stuff like this. But it really goes beyond that. There's so much meeting and organizing and working with a team to get these things done, to move things forward. You're not from. Did you grow up in Winnipeg? You didn't grow up in Winnipeg, did you? How'd you end up here?
No, I'm an American. I was born outside New York City in New Jersey. Most of my family lives there. I also spent 15 years in Chicago, so I still have friends there. Don't have any relatives there anymore. And I lived in Cleveland before coming here. I was at the Cleveland Clinic for 4 years in Cleveland, Ohio. So we're new to Winnipeg, so to speak, 15 years later. I have 4 daughters, and my youngest daughter was born here, right here at St. Boniface Hospital.
Sure. So yeah, and that's another wonderful part about what we get to do here is there's so much you can move and share and spread what you've learned around the world. So your lab, like we said, kind of looks at nerve function and memory. I always say to kids that I work with is memory disease research must be super challenging because we don't really understand how memory works in the first place, right? It's not like a heart that just we understand how it pumps blood, and when heart disease happens, we can study it. But it must be really challenging to study diseases of the memory. Like what's your big goal in your lab? What's kind of the big question you're trying to answer in the work that your lab does?
Well, that's an excellent question, and I think you're right. It is challenging, and it's challenging for a variety of reasons. If you look at the history of neuroscience research, in the old days, a lot of this stuff was very compartmentalized. You had the psychologists working on behavior. You had the biologists working on the organelles and energy and all this stuff. And, there's a famous scientist, he's still alive, he's at Columbia University, Eric Kandel, who kind of brought together disciplines of psychology and biology. And he received a Nobel Prize for his work with synaptic plasticity and a protein called CREB, a transcription factor. And it was his work on CREB and the sea slug that really kind of highlighted how the brain function and synapses drove certain behaviors. And so that's kind of set the stage for really the whole area of neuroscience that we have. When I was working on my bachelor's degree, they didn't really have neuroscience then as a degree choice. I had to form my own major. I got a bachelor's in general science. And so I took biology, I took psychology, I took physics, I took a little chemistry and math. And all those things together is really what makes up neuroscience. It's an interdisciplinary approach. And so because of that, we can study complex things like the brain. And so, like you said, it's an incredibly challenging thing, not only scientifically, but other aspects, just getting funding, getting donations, just trying to get the word across and making it easy for people to understand. And that's one of the things that's very important is really, we can talk in techno speak all the time and use jargon that nobody understands, but that's not what this is all about. And tax dollars are there to help bring information to society. And so that's pretty cool, is that we can do stuff in the lab and try to make sense out of it and explain it to you folks that are watching right now.
Yeah, and so a lot of the work, I mean, I follow along a little bit with some of the work that you do, and I know you kind of go from the whole organism and how memory can work all the way down to really just what's going on in those individual little nerve cells or neurons. And I'm always fascinated because, I've been at this a while too, and I get to talk to all kinds of people, and there's always things that really interest me that pop out of labs like yours or other things around here that I think I never even thought about it in that way. And so you'd shared one of the publications from your lab with me, just something. This one's, this will probably confuse some of our listeners out there, but mechanisms of mitochondrial dysfunction in Alzheimer's disease. And this is one that really interested me because I think if we have some grade 8 students out there right now watching this, they've probably heard of the mitochondria. They've probably labeled a cell diagram, and you can label your nucleus and your mitochondria and your other things. And they could probably all tell us that the mitochondria, is the powerhouse of the cell. And, that's what I learned too. And you just think, okay, well, it gives energy to the cell. But when I read things like this, like some of the things that your lab is learning and pulling together from other scientists, you really realize, oh, there's way more to some of these things, and they're really involved in disease. Now, this one looks at, here we're talking about Alzheimer's disease and how those mitochondria are involved in some of those nerve cells. Can you explain, just before we dive into the mitochondria science, when we say Alzheimer's, people are probably familiar with this as a disease that affects memory. Is it part, and we use these words dementia and Alzheimer's that our listeners might be, or viewers might be familiar with. What's the difference when we talk dementia, Alzheimer's? Can you explain that just a little bit for us before we drill down?
Yeah, so I've been, like I said, studying memory for a long time, and I've been studying Alzheimer's disease for about 15 years, and I have actually a special connection to this because my mother died from dementia. So Alzheimer's disease is a type of dementia. It's the most common form of dementia. Scientists define dementia as a neurocognitive disorder, but originally when people used the word dementia, what it meant is that it means without mind or almost insane. And it's kind of an unfortunate use of the word or term because it really carries a stigma. So it is a neurocognitive disorder. And if one of the hallmarks of Alzheimer's disease is that the memory loss. And many of us have family members that have been affected for Alzheimer's disease. So this is really shouldn't come as any surprise. But for Alzheimer's disease, one of the classic symptoms is the memory loss. Then there are other forms of dementia, though. So there's Lewy body dementia and Parkinson's disease dementia, and a variety of different types. And these don't all actually have memory loss as one of the symptoms. So there's also hallucinations. Some people might not actually have memory loss. They might have problems adding and subtracting or with other executive functions. And so it just depends. So, but with Alzheimer's disease, that is what we focus on in the lab. And like I said, when my mom developed dementia, it really changed my focus scientifically and personally because we have to get the word out. We have to try to get people to understand what it is and what it isn't. Mm-hmm. It isn't we just don't all get Alzheimer's. And just because we get older doesn't mean aging leads to Alzheimer's. There's a connection, but it doesn't mean they're not the same. Aging and dementia or aging and Alzheimer's disease aren't the same. So, we've got to educate the public about these differences and also the treatments and what we can do to prevent Alzheimer's disease, such as exercise and diet and keeping our blood pressure low and lots of different things out there that we actually have control over.
Right. And so that's a big part of the work, in a research center like ours is we've got to try and get right down to what's actually changing inside the brain or what's changing inside the heart to cause heart disease. What's actually changing all the way down at that cellular level. And that's kind of where your lab spends a great deal of its time as I look through, pictures like this, or papers like this, we can see really interesting diagrams that show us how neurons change
Yeah.
You can have a healthy neuron, which, you know, a lot of our viewers out there, their brains packed full of nice, healthy neurons that can do their job every single day. When you start looking at those nerve cells out of Alzheimer's brains, they change. Can you walk us through a little bit? I know you've got some slides prepared too. Walk us through a little bit about, you know, how you try and study those changes. What are some of those changes that we see?
Yeah, so those are great questions, and there are a lot of things going on. There are a lot of moving parts, and this is part of the reason why it's so difficult to develop drugs that are truly effective or highly effective. And so, what people have studied for 25 years or more are these so-called plaques, which are sticky proteins that contain something called amyloid beta, which in the most part is toxic, and then tangles. So different filaments that get tangled up and impaired transport of materials in the cell, kind of like if somebody had a roadblock on a major highway, transport is disrupted. And this is what happens. These are the two classic hallmarks of Alzheimer's disease are the amyloid plaques and the sticky or the tangles. But there's lots of other things that people are studying. Like my lab has been looking at mitochondrial function for several years now and what role it might play in Alzheimer's disease. So, you know, when you study the mitochondria, really what you're interested in is energy and energy metabolism. So another way of looking at it is to think about, you know, do you have the energy to run your house? Do you have a furnace? Do you get heat from your furnace? Is it working properly? Or when you have a flashlight, you have batteries in your flashlight, you can tell pretty quickly when that flashlight is dying or the batteries are going weak because the light's getting dimmer. Well, mitochondria are very similar. As we age or in certain diseases, the mitochondria aren't functioning correctly anymore, and we don't get as much energy output. So this is one of the things we really focus on in my lab, and we look at a lot of different things. So, you know, I'm very proud of our lab because we have a lot of different methods that we use to study all, not just proteins and molecules, but also behavior. So we use imaging to look at the brain. We use behavioral tests to do memory testing in both animals and humans. We study proteins using cell and molecular methods, and there are a lot of different approaches for doing cell and molecular tests. We do something called electrophysiology. We'll measure electrical activity in brain cells and in brains. You can do this in the cell, or you can do it actually in the whole brain. It's kind of like an EEG. So, you know, it's good to use lots of different methods because you get the big picture then.
Yeah.
And by taking these approaches and integrating the information, we can get a better idea of what's going on with the proteins and with behavior and with the DNA to really tell us, you know, give us a complete picture how memory is working and how it's getting impaired and how those impairments lead to disease. So, you know, we start with the DNA, then we go to a protein, and then we go to an organelle, and then we start to see how cells start to die. And once the cells die, then circuits start to fall off. And when the circuits, the neural networks aren't working, that's when you start to see changes in behavior. So
Sure.
it's all connected in very interesting ways.
So I think you had some pretty pictures to show us there. Do you mind sharing some slides, some
Sure.
stuff from your lab
Let me
to
show you
kinda
a couple of
show
slides
people
here.
what we're talking about?
So these are just a few images. Can you see that okay?
Yep, that's coming through just fine.
Okay. So just to kind of reiterate what we were just talking about, what causes Alzheimer's disease? Well, for 25, 30 years, people were looking at the plaques and tangles, and we know now that the plaques and tangles are important, but it doesn't tell the whole story. And so that's why our lab is looking at energy metabolism, mitochondrial function, and other proteins like transcription factors and signaling pathways.
And so in that picture,
just,
can you go back for me there, Ben? I just explain to our viewers, like, so in this picture, like these purple ones here, like those are our nerve cells, like our actual neurons, and they really need to be able to communicate with each other, right? Like, and when we get these blobs of protein, these plaques and these tangles, they really can't do the communicating that they need to do. And if you think about how your memory works, those nerve cells has to be communicating all the time, and anything that kind of gets in the way is going to affect how your memory works or anything else, right?
Well, that you hit it on the nail on the head, Steve. And it's, you know, there's two main communication systems in the body. It's the nervous system and the endocrine system, or hormones. And so, so what we're looking at in this slide, those purple triangular things are cells. Those are nerve cells. And those nerve cells get screwed up in many of these diseases, especially, or in dementia, especially in Alzheimer's disease.
Great.
So this other slide, I just wanted to show you real quick. We talked about the mitochondria, and you can see in the cartoon on the left is an illustration of the mitochondria. So this is really where the energy is generated, and it's generated in the form of ATP. So it's really the energy currency of the cell, and it's this ATP that we study in the lab in part. And we use very sophisticated equipment and instrumentation to measure ATP, and we can do this in different ways. And we can do it actually in living cells. So, and we're going to start hopefully doing this in human cells, in human white blood cells soon too, and in some of our clinical trials that we're proposing in the future. Cool. So we're very excited about this work. And the other thing to notice in this slide is if you look at mitochondria, it plays a huge role, not just in Alzheimer's, but you can see in Parkinson's and heart disease and diabetes, aging, and so on. So it's really central to so many different diseases and conditions. And this is one of the reasons why I really get excited about our research here, is because if we make a breakthrough with mitochondrial dysfunction, we might not just solve important problems for Alzheimer's disease, but we might have answers for other scientific communities as well.
Because energy is important to any cell, right? I mean, your brain is super energy hungry. It needs tons and tons and tons of energy to do all the things it does. But every cell needs a steady, constant flow of energy to do its job.
Especially the brain.
Sure.
And the brain has a very high energy demand, and because of that, we have to keep it supplied with glucose and oxygen. Otherwise, we pass out, and then we have serious problems.
Mm-hmm. So what are we looking at here?
Well, here we have a series of PET scans that we did. In fact, this is not just PET scanning, but it's an overlay of PET scanning and MRI. And MRI stands for magnetic resonance imaging. And so this is pretty cool, because what we do here is that we use two different instruments. They're combined. And so the PET scanning allows you to measure function. And you can see functional changes in these blue shaded areas. So those blue shaded areas in the image here are areas that are hypometabolic. They're not getting enough glucose. They're not getting enough sugar to run those regions of the brain properly.
So that's actually showing you, just for like PET scanning is a technique where we can actually look at where that sugar, like the glucose, is being burned. Like that's really where the energy.
Glucose uptake, yeah. So we use something called a tracer that helps to follow molecules through the brain as they're being metabolized, and that's exactly what we're doing here.
And here, just for those viewing too, like these, it's almost like we're looking at slices, right? So it's almost as if you've almost as if you've taken my brain and you've started somewhere here and you've done a slice and 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 a slice. And then we're looking at those slices. Are we looking from the top? Am I right on that?
Yeah, so these are called horizontal sections, and they're in a mouse. And so sometimes the anatomical terminology is a little different for the human. But yeah, we're looking at a variety of, so we're going from top to bottom as we go left to right, and as we go from the first row, the upper row, to the bottom row. And so we walk through a series of slices, even though it's a whole brain, but you know, for using this imaging modality, it looks like we've sliced the brain, but we haven't.
Right, it's just a machine like you or I would go into at the hospital if they needed to get a scan of our brain and see how our brains were functioning.
That's correct. And so again, those blue regions are regions that were created by using the PET scanning instrument. And then the other region that shows this really high definition anatomy is the MRI.
Oh, okay.
And so by putting them together, you get a really powerful scan or image that's formed. And so what we do in this is that we were able to identify those regions that are susceptible to Alzheimer's disease, because this is actually a transgenic mouse model, a model that's been genetically engineered to have the mutations that one sees sometimes, not all the time, but sometimes in Alzheimer's disease pathology. And so what we'll do then is that we'll further test samples from those blue regions in many of our other methods, our cell molecular methods in particular.
Cool. Yeah, so that's going from the whole organ. There's changes you can see at the whole brain level, but then you can break it right down to the cellular level as well, what's going on in individual cells.
That's correct, and I think that's the approach we have to take. We start with the behavior, we start with the whole person or a whole animal, and then we work down. So in this case, it's a top-down approach. Now here, I'm going to show something, and again, this is a nerve cell, but what we're going to see is, and we're looking at not just a nerve cell, but we're going to look at these organelles that we talked about just a few seconds ago that are called mitochondria. And this video was supplied by a postdoctoral fellow here at our hospital research center, Dr. Sabir. He created this. He needs to get the credit for this, but it's a movie that he made that traces the mitochondria as they move around.
Cool.
Tell me if you can see this in real time.
Yeah, that's working just fine. Amazing.
Okay.
Yeah.
So you can see that the mitochondria have been labeled with this red fluorescent compound. So you can actually physically see them moving around in the cell, and in this case, it's a nerve cell.
So they almost look like as if you're looking at highways from above, right? Like it's almost like little cars on a highway. And are you telling me like those are the red that we're seeing, like that's actual mitochondria running down these kind of highways in the nerve cell?
Well, that's such a good analogy, Steve, because that's exactly what's going on. And what we said a few minutes ago is that the tangles that we see, when those get disrupted and when they get chemically altered, it's the highway system that gets screwed up. And we can physically see that in real time, and we can visualize that in an experiment like this. This is actually called confocal microscopy. So you're exactly right, is that these mitochondria move around the cell, but what they do, importantly, is that they move to sites of high energy demand, because the memory and the brain require lots of energy all the time. And if you don't get mitochondria moving to those sites of high energy demand, you're not going to be able to think and feel properly, and you might even lose consciousness. So not only is glucose uptake important, but also mitochondria delivering ATP and actually even another atom called calcium. So these are delivered just like a truck would be delivering supplies to another part of the city or another province or state and so on.
Amazing. That's some really beautiful. It's almost artwork, isn't it? I've always loved Sabir's. Sabir's great at taking pictures under the microscope, and I find some of his pictures are almost artistic. Like they really have a lot of interesting creative qualities to them. It's really beautiful to see.
And I mean, the other thing to appreciate here is that there's a whole nother world going on. It's like world within worlds, right? So we have a whole nother universe going on at the molecular level that we can't really sense directly, but it's all going on. And like you say, it's like a highway system, and there's all kinds of activities going on. Cool. So that's about all I want to show you today, but I certainly want to give credit to my staff. They're not all in this photo, but I list them all here by name on the left. And so postdoctoral fellows like Dr. Wanda Snow and Ida Adlimigodam and Yela Dvorževic, and also Don Davies, that are all really project leaders, and then a variety of other technical support like Claudia and Nancy Olson and other students that we have in the summer and throughout the year. So all of these people contribute greatly to our success here at St. Boniface and our research program. So I just want to give a thanks to them as well.
Yeah, it really is a team effort, isn't it? And I think we often people think, oh, there's a scientist, one guy doing his job, but it really is a team effort of everybody working together on little pieces of a bigger problem that you get to kind of direct and figure, okay, what do we want to do next? But it really is a team effort every step of the way.
It is a team effort, and I have to tell you, St. Boniface is such a great place to work, and it's people like yourself and Grant Pierce that have made it a place that a team can work at. And so it's a great place for training. It's a great place for publication. It's a great social atmosphere. Sure. I mean, we have great parties here when we don't have a pandemic going on, right?
Yeah. I see Angelica's giving me a wave there. That must mean we have a question in the chat.
Okay.
In Alzheimer's, do the plaques and tangles cause the neurons and mitochondria to change, or do the mitochondria and neurons change, and that causes the plaques and tangles to form?
So I don't know if you could hear, but Angelica's question was kind of a chicken and egg question. Is it the plaques and tangles that are causing the changes in the mitochondria? Is it changes in the mitochondria that are causing the plaques and tangles? Is it one before the other? Is it like how does it all go together?
Well, we believe order matters, and that's a great question. We can't answer all those questions right now, but I can tell you that it seems the data suggests that the plaques form first. And then once the plaques start forming, and this is a long process. One thing we didn't mention is that Alzheimer's disease probably takes the course or span of 20 years or more. So these are slow changes. They don't happen overnight, but it does seem like the plaques form first. And then we get changes, we get chemical alterations in tau and the disruption of transport systems. That comes later. But with regard to the mitochondria, we see our mitochondrial changes much earlier than these big plaques that are being distributed. However, what I didn't tell you is that plaques have a component. They have several components, but one of the main constituents of the amyloid plaque is amyloid beta. And so this particular molecule that forms the plaque, we see accumulating in the mitochondria. And so we believe right now, anyway, that it's the accumulation of it's the accumulation of the amyloid beta in the mitochondria that disrupts different mechanisms in the mitochondria. And those are some of the things that we're trying to investigate. So we don't have a clear answer yet, but that's certainly a direction that we're looking at or following right now.
And I think that's a really important thing that in all of this is going to come up in every single one of these that we do is like, we just don't have a clear answer yet, right? And this is we've got to just try and figure it every step of the way. I'm no Alzheimer's expert, but when I hear you say like, this could be something that happens over 20 years, does that mean like that maybe work like yours or work from other Alzheimer's labs, maybe there's things that we could detect in brains even before people start losing their memory? Like, is that kind of is that one of the things you're hoping to find? Like that we can maybe figure out this is happening before it gets to the point where memory doesn't function?
Yes, so to that end, there are several things that are in recent development. In fact, some of the meetings I've been to this year have released new data. And so it's not just the brain, but the brain is part of it. So for example, there's specialized types of neuroimaging that are focused just on imaging amyloid or focused on imaging tau. And so those specialized neuroimaging modalities have become an important biomarker and part of the process. But also now we have new blood tests. And some of these blood tests have really improved in their predictability and accuracy. So we can take some of these, get someone's blood and start to measure biomarkers in the blood, and tell much earlier with better accuracy that a person is at risk for transitioning to more severe stages of Alzheimer's.
Cool. Well, I think we can wrap it up pretty soon. I mean, Ben, you're a great friend of our lab. You're always so open and willing to share, and same with the rest of your team. I know we've had a bunch of folks from your lab down here sharing with students, and we always greatly appreciate your time, your passion, and your interest in what you do. It comes through every single time, and I hope people see that. Even if you didn't understand all the big words out there, this is what we really want you to take away from some of these chats. It's really just about being curious and being creative in how you approach these really complicated problems. Does that do you find creativity is a part of your work in your daily life?
Well, for me it is, and I think not everyone thinks that way. I mean, and people have different skill sets, and there's multiple intelligence theory for the way your mind works and this sort of thing. But yeah, you don't have to be a mathematician. You don't have to be a computer scientist, although it helps. You can have other interests. You might be good at visualizing things. You might be good at equipment or interacting with technology. So there's a lot of different roles to play in medicine and in science that you should be aware of. And creativity, I think, is really overlooked. I mean, I think we talk a lot about critical thinking as we train students, but I think we overlook the importance of creative thinking. Yeah. And I think if you look at all the great geniuses and some of the scientific discoveries over the years, there's been a really important creative element to a discovery, a dream or some sort of aha moment or an insight that really kind of advanced science at that moment in time.
Yeah. Well, that's fantastic. Again, thanks as always for sharing with us. I'm sure a ton of people have learned a ton today. And they're always welcome. Make sure follow the Youth BIOlab on Twitter and Instagram. Follow Dr. Ben Albensi at BIOlab on Twitter as well. He's always putting out content. And always interesting to see what his lab is up to. Join us again next week. I think it sounds like we've got Laurie Kirschenbaum, Dr. Laurie Kirschenbaum from the Institute of Cardiovascular Sciences joining us to give us his share his perspectives on heart disease. Ben, maybe you can encourage some other folks from your lab to join us sometime. There's always a million more questions. That's, as science goes, it's always you start with one question and it just leads to more and more and more and more. I know you're always into lots of interesting things. Thank you very much for joining us. We'll see you again soon.
Okay, thank you, Steve. Thanks for having me.
All
Stay
right.
well.
Yep, thanks.